Actually, today just I will give you a glimpse of the windows to the nervous system. Just I will. It will be a rather a slideshow than explaining things because the basic the skull has to be completed. Along with this, you will be knowing because I thought of showing the video, but you can take it from the demo room in your pen drive. Now, window to the nervous system. Window to the nervous system is a journey through the nervous system. I don't know if I will be able to complete it or not. Now, tell me where is fancy bread, in the heart or in the head? This is a quotation from Shakespeare. Tell me where is fancy bread, in the heart or in the head? Shakespeare. Now, although Shakespeare has asked this question near the end of the 10th century, the answer had been known at least to some for more than 2 millennia. 2 millennia. Many ancient Greek scholars, Hippocrates and Plato, among the appreciable that there is something special about the brain and argued that the brain is responsible for behavior in humans and other animals. This is the basic thing you have to know, the behavior and and we know now take it for granted that the brain is the organ that obtains information about the environment, processes and stores this information and generates behavior. In addition, it is the brain that is responsible for such vaguely defined aspects of behavior as feeling, aspirations, and abstract thought. Those qualities we consider with more than a touch of hubris quite essentially human. A feeling, aspiration, expectation, these are actually human feelings. Even animals, even the plant kingdom, if you go to the thing that plant has life, Acharya Jagadish Chandra Bose's experiment, and now the diseases of the brain, which often are associated with aging and hence are on the increase as the human lifespan lengthens, cause untold suffering for individual families and society as a whole. Now, Alzheimer's disease. Now, this is a very the condition of the brain. A perfect person around the age of 75 he gets an Alzheimer's, and everything is the painful to the society and also the painful to the near and dear ones, the family. So, in essence, then it is the brain that makes up what we are, and indeed it can be argued that all other organs are there simply to support the brain. For this reason understanding the biology of the brain function is a major goal of modern science. The brain is one of the last great frontiers in the biological science, and the unveiling the mysteries is comparable to complexity and intellectual challenge to the search for the elementary particles of matter or the effort to explore space. Now, these are the human embryos of different yard length. You can see the smallest one is here, and then gradually you can see the fetal thing. The fetus of different yard length. Now, this is a collection, my photograph. and it is actually, you won't be finding it any embryology book this size, and this had been collected more than 20 years ago. Now I have dissected cerebrum in human fetuses. Now the fetal skull, you can say cartilaginous, they were dissected out, and the two parts of the brain, you can see the brain over here, sulci and gyri. This is the ventricle that is exposed, I'm showing, and this is the fourth ventricle. You will be knowing that today you will just see the dissected brain and spinal cord of the human fetuses. If you see, actually, the 35th gray, I got the inspiration from 35th gray, that is a 35th edition. That brain and the spinal cord in a blue background, a total dissected in a adult. So I presented a paper on sulci, appearance of sulci and gyri on the human fetuses. This is the brain and the spinal cord. Now areas of the brain, you can see the prefrontal cortex, Broca's area, just you'll be knowing after a month. You go to, after head neck, you will be motor cortex and parietal lobe, gustatory areas, ventral area, primary cortex, optic radiation, cerebellum, brainstem, this is the brainstem, primary auditory cortex, left cerebral hemisphere and left middle cerebral artery. Now, there is a beautiful illustration in Gray's anatomy with a man putting a hand on the brain. You can see it's a very beautiful illustration. Please take the photograph if you don't have possess a Gray and keep it. It's nice to look at and also very, very, very informative. Now the major external parts of the human brain, underside view. Now the frontal lobe, olfactory bulb, temporal lobe, pons, cerebellum. Now this is the cerebellum gross anatomy. Now location of cerebellum, you can see the location, and I told you about the skull, frontal bone and parietal bone when I was demonstrating the sinuses. Here are the sinuses. A pair of cerebral hemispheres united by the vermis, worm shape. This is the anatomy of the cerebellum. Now these are the connections of the cerebellar peduncles. You will get it in your textbooks. Now the classification by phylogenetic and ontogenetic development, the archae the archicerebellum, paliocerebellum, and neocerebellum, classified by afferent connection, vestibulocerebellum, spinocerebellum, pontocerebellum are classified by efferent connection, permis, paraphragmal region, cerebral, cerebral hemisphere, these deep cerebellar nuclei, you will see the, you will be given the brain, you can see the specimen, cerebellar peduncles, the blood supply of the cerebellum is the superior cerebellar artery, anterior cerebellar artery, posterior inferior cerebellar artery, venous drainage, superior cerebellar veins drain into the straight sinus, great cerebral vein drain transverse and superior petrosal sinus. Now, the veins drain into the sinuses of the now functions of the proprioceptive sensation is now you can see the cerebral is the head ganglion of the proprioceptive system. You will read in physiology, and since it receives impulses from the muscles of the limb, trunk, and face, etc., it controls the tone and posture and equilibrium. It is concerned with unconscious impulses, especially kinesthetic and vestibular, which controls tone and posture and equilibrium. Now, Bidan Rai ba oi those great clinicians and physicians of yesterday, even today, even if you can, they used to say, Bidan Babu দেখলেন যে একটা লোক এরকম করে হাঁটছে, হ্যাঁ, দেখেই বলে দিল যে তার এই ব্রেনের ডিসঅর্ডার হয়েছে, সেরিবেলাম ডিসঅর্ডার। এগুলো মানে ট্রু, অলসো ইট ইজ the they had so much clinical knowledge that most of the things you can diagnose that which part of the brain is diseased. Functions of the cerebellum, it guides the motor cortex and controls all voluntary movements so that they may be accurate in time, force, duration, and extent. It exercises all kinds of final editorial supervision over the motor impulses emerging from the cerebral cortex. Cerebral cortex. Now, this is I like this word sentence. It exercises all kinds of final editorial supervision of the motor impulses. The editor of a newspaper or editor of a news channel in the television, they get the information, but they will select and give you which is to be presented. Similarly, so cerebellum has a great function in functions of cerebellum. It exerts synergistic control. The control of cerebellum is ipsilateral, and this you will be knowing when you go further. Now, the cerebellar ataxia. Ataxic gait and position left cerebellar tumor, sways to the right in standing position, steady on the right leg, unsteady on the left leg, ataxic gait. Now, this is a clinical experience we will see patients in the outdoor of your MBBS course. আর তোমাকে খুঁজে বেড়াতে হবে মানে একটা লোক হাঁটছে, তুমি তখন সেই সময় যাচ্ছ হসপিটালের ভেতর দিয়ে, ওয়ার্ডের ভেতর দিয়ে, তো একটা লোক এরকম করে হাঁটছে, তখন তুমি দেখে নিলে যে না এই পেশেন্টের এই হয়েছে, হ্যাঁ, বা সে আসছে, তোমাকে রিলেটিভ বলছে, রিলেটিভ ইজ সেইং দ্যাট বাবু এইরকম হাঁটতে লেগেছে এই কদিন থেকে, তখন তোমার মাথায় আসবে সিটি স্ক্যান, এমআরআই, তারপরে তোমার nerve conduction velocity test, all these tests which will be available in the medical college, so you can prescribe for your own thirst of knowledge and also to present this with your, you will gain the clinical experience. Now, cerebellar lesion, ataxia, hydrocephalus, hypotonia, এগুলো যত আসবে you will be knowing nystagmus, intense tremor. Now, nystagmus, the eyes display oscillatory movement when they are fixed to an object. The eyes tend to deviate from the control position to 10 to 30 degrees to the opposite side. এটা কিন্তু তুমি দেখবে টর্চ দিয়ে একটা ওয়ার্ডে একটা পেশেন্ট এসছে, সেখানে তুমি you see if she is having or he is having the nystagmus. It's an oscillatory movement. Don't বলে চোখটা fixed রাখুন. You please keep your eyes fixed. Then there will be oscillatory movement. Now cerebellar tumor. Now the cranial nerves. Now olfactory, optic, oculomotor, trochlea. This is first nerve, second nerve, third nerve, fourth trochlear nerve, fifth trigeminal, sixth abducens, seventh facial, eighth vestibulocochlear, ninth glossopharyngeal, 10th vagus, 11th accessory, and 12th hypoglossal. Now, the 12 cranial nerves you will be dissecting. It will be demonstrated to you when you dissect the neck, head and neck. You dissect. You will see all these nerves in your dissection in the dead body. It is very clearly shown, and you can identify fish mota mota. I'm using this word mota mota. So facial nerve এরকম ব্রাঞ্চিং দেখতে পাবে, প্যারোটিডকে পেনিট্রেট করে আসছে, এদিকে glossopharyngeal nerve. Every nerve you will be identifying in your dissection. Now cranial nerve nuclei, you can see the undersurface of the brain. We have specimens there. You can see individual, don't tamper the thing. You just take your pointer or forceps when you put the brain, you turn it up and see all these cranial nerve nuclei. Now cranial nerves, this is a very good illustration showing each and individual where it is supplying and what organs. Now olfactory nerve, optic nerve. Now there can be a question on optic nerve, the optic chiasma, and it is essential. Now trigeminal nerve, ophthalmic nerve, maxillary nerve, and mandibular nerve. Facial nerve, you can see the branching. Now, again, a question can come, describe the facial nerve, the origin and supplying the respective organs and the muscles. Vestibulocochlear nerve. You can see this is the vestibulocochlear nerve here. This is the cochlea. These are the ossicles, glossopharyngeal. Now, it is the brainstem. Now it supplies the tympanic membrane, the tonsils, glossopharyngeal nerve here, and supplying the tongue and the carotid artery. Now vagus nerve. Now you are acquainted with the vagus nerve in the thorax and hypoglossal. Here's the hypoglossal nerve. This is the tongue. This is the internal jugular. This is the face. This is the lingual vein, lingual artery in red, and veins of the dorsum of the tongue. And this yellow is the hypoglossal nerve. Now, cracks of the spinal cord. I'm just giving you a glimpse of the receptors. The ascending tract. This is the ascending tract, and the cerebral cortex is going here. Lateral spinothalamic tract. As you progress in your journey of the brain, the nervous system, you will be acquainted with this type of illustrations and pictures and description. The anterior spinothalamic tract, touch and pressure crudely localized. Posterior spinocerebellar tract. Posterior spinocerebellar tract. The third lumbar vertebra, muscle, spindle, Golgi tendon organ, joints, receptors from trunk and lower limb, unconscious proprioceptive, touch, pressure, posture and movement. এখানে কোনো একটা ইনজুরি হয়ে গেল স্পাইনাল কর্ডে, তো these sensations or the actions of the nerves relating will be lost. So you can diagnose that at this level the injury is there. Posterior columnar tract. Again, posterior columnar tract has key function, proprioception, vibration, fine touch, two patient point discrimination, tactile localization and stray-ness. এবার তুমি বসে আছো একটা কোণে, হঠাৎ করে লোডশেডিং হয়ে গেল, তুমি কিন্তু আস্তে 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 গিয়ে সেই তোমার টেবিলে গিয়ে তুমি ঠিক ঘড়িটাও তুলতে পারবে, পেনটাও তুলতে পারবে। দ্যাট ইজ stray-ness. Tactile location, 2-point discrimination, 2-point discrimination. Now, corticospinal tract, skillful voluntary movement of the postural type, speed and agility, modulate sensory input, smooth performance of movement, flexor muscles of the distal parts. Just be acquainted with the names, corticospinal tract, rubrospinal tract, vestibulospinal tract, is the vestibular nerve here and lateral vestibular nucleus. Reticulospinal tract, tectospinal tract, olivospinal tract, lesions of the spinal cord injuries, traumatic accidents, gunshot, fall, stab wounds, fractures, dislocations, ligamental tear. Non-traumatic, disc prolapse, infections, abscess formation, vertebral subluxation, and spinal neoplasm. Now, non-traumatic disc prolapse, you are carrying some weight, your backpack, and you are traveling in Shantiniketan Express today, or not today, Shantiniketan Express, you go by Shohid or who is good traveling to Calcutta, you not got a seat or reservation. So carrying weight and don't pack up all things. That way you have to take care of your disc, your own disc, and the accidental injuries. Complete transaction above third cervical segment, paralysis of respiratory muscles, unable to survive. Below fifth cervical segment, quadriplegia and paraplegia, complete loss of all sensation and voluntary movement. Now, a third cervical, fifth cervical segment যখন একটা পেশেন্ট তোমার ফিফথ সার্ভাইকাল সেগমেন্ট নিয়ে তোমার কাছে এলো, তখন তোমাকে বলে দিতে ভাই অপারেশন টপারেশন করে বিরাট কিছু একটা ফায়দা হবে না। আর যদি ইমিডিয়েটলি আসে সেখানে দেখতে হবে যে কতটা তুমি রিকভার করাতে পারবে। বাট ইউ unnecessarily don't do a surgery or think of doing a surgery because you won't get the result. আর একটা হতে পারে তুমি অপারেশন করলে you come and tell the patient party that operation is very successful but the patient is dead. So এটা কর্পোরেটের এখন খেলা চলছে, বাট একটু মূল্যবোধ, একটু হিউম্যান ফিলিং ইউ হ্যাভ টু হ্যাভ। তোমার নিজের মা বাবার যদি এটা হতো তাহলে কি তোমার আমি একজন নিউরোসার্জন, তোমরা কি বলতে স্যার এটা তাহলে অপারেশন করে দিন, আর আমি বলতো হ্যাঁ হসপিটাল অ্যাপোলো হসপিটালে ভর্তি করে দাও, আমি করে দিচ্ছি। নো 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 নো। The incomplete lesions, anterior cord syndrome, posterior cord syndrome, central cord syndrome, Brown cord syndrome. These are the lesions of the spinal cord. Anterior cord section injuries, dislocation, disc, wedge fracture of vertebral body, contusion, injury of the arterial spinal artery, C4, C7, C12, T12 and L2. Now, rare condition, posterior cord syndrome is a rare condition, fracture of the posterior elements and avulsion fracture. Central cord syndrome, hyperextension injuries, whiplash injuries, congenital or degenerative narrowing of spinal cord, more severe involvement of upper extremity and sacral sparing. Brown cord syndrome, hemisexual spinal cord, stab wound, compression due to extramedullary tumors on the side of lesion, damage of posterior funiculus, pyramidal tract. On the contralateral side, damage to the lateral spinothalamic tract. Now, in United States of America, they have surveyed that most of the MRI, CT and CT itself for brain lesions, actually that could have been diagnosed clinically, but things have been done at random. The MRI, CT or CT, actually you don't, most of the neurological examination, it can be with your hammer, with a needle, with a feather. A tinte instrument and your clinical knowledge, you can diagnose more than 80%, 85% of the diseases of the neurosciences. Upper motor neural lesion, you'll be reading in physiology also, these lower motor neural lesion, pharyngomelia, milia, cavitation and glossus in the central canal, cervical region, pharyngomelia, clinical manifestation of spinal cord lesions, এগুলো you will be spinal shock, এগুলো সব তুমি দেখতে পাবে, এগুলো পরপর পরপর। I won't overload or crowd in your brain. Now blood supply of the brain, internal carotid, circle of Willis. This is that a question can come from circle of Willis. Okay. Anterior, middle, and posterior cerebral arteries are give rise to two sets of branches, cortical and central. Central branches consist of 6 vein groups: anteromedial, posteromedial, right and left anterolateral, right and left posterolateral. Cerebral cortex supplied by cortical branches of the anterior cerebral artery, middle cerebral artery, and posterior cerebral artery. This is an important thing, the arterial supply of the internal capsule. The internal capsule of the brain are often asked in your written paper, and if you are answering for honors or gold medal, then the examiner will question you on these levels. Artery supplying of the thalamus, artery supplying the corpus luteum, artery supplying the brainstem and cerebellum, veins of the brain, veins of the cerebral hemisphere, cerebellar veins, and veins of the brainstem. The cerebral veins are divisible into external group and internal group. Internal cerebral vein, basal vein, and great cerebral vein. Veins of the brainstem, midbrain drains into the cerebral vein, pons upper part into basal vein, medulla oblongata into transverse and occipital sinuses. The medulla oblongata does not drain into the vein but transverse and occipital sinuses. You can see the location of the medulla oblongata, the occipital lobe, veins of the cerebellum, dural venous plexuses. That can be a question on dural venous plexuses. Meninges of the brain. This is the skull, the neuromata, periosteal layer, meningeal layer. Arachnoid matter, blood vessels are here, pia matter. The brain, it covers the brain. Ventricles of the brain, third ventricle of the brain, lateral ventricles of the brain, now this is the normal brain section and hydrocephalic brain. You can see the ventricles. Brainstem composed of midbrain, pons, medulla oblongata. Brainstem structures, spinal cord, the spinal cord has an importance in anesthesia and also for lumbar puncture in to get the CSF fluid in meningitis or your encephalitis. This is spinal cord internal structures, the anterior median fissure, posterior median septum, central canal in the center, grey matter in the middle, and white matter in the periphery. Spinal cord proper. You can see the thoracic nerves, the lumbar nerves, sacral and coccygeal nerves. Spinal cord internal structures you will be seeing in histology. Internal structure of the spinal cord, internal structures, white matter, arranged in columns, tracts, ascending sensory tracts, descending motor tracts, anterior lateral cortical spinal tracts. Now spinal cord cross section. Now flexion rotation injury. If this type of a man carrying a load and suddenly he slips and you see the type of injury he is sustaining, sustained to. If vertebral fractures are there, crush injury, একবারে ডিগবাজি খেয়ে পড়ে গেছে, and you see the pattern of the vertebral কি অবস্থা হয়েছে, একবারে flexion injury. Any game you are playing, you can get prone to such type of fall. You get a flexion injury. Extension when you're driving a car, head-on collision, and you can see this driver head-on collision. Now, chance fracture. You're sitting in a seat. Suddenly there's a jolt, and you get a fracture. This is the fracture you're getting here. This line. Articulation of the facet joint in this region. Atlantoaxial injuries. Tuberculosis of the spine and cold abscess. This is a very common lesion in our country. The tuberculosis of the spine. And a cold abscess should never be drained, because then it will be continually, you have to treat, diagnose, treat, and treat with medicines. Then, and of course, some splints and supports to be given. This is spinal cord tumor. X-ray of the spine. The MRI of spine, you can see here the area of lesion. The MRI picture. Now, these 2 days you enjoy, relax, and again come back to the last lap of the marathon. Wish you all the best. You will be seeing things in the dissection hall. All specimens are there, and you have to just open your eyes and see as the classes progress. You just
some of the tools of recombinant DNA technology. Now, all of you know that what do you mean by restriction endonuclease? If I ask you, you can answer something. Yeah? Can you answer something? Molecular scissors, very good. Used for cutting DNAs. DNA or RNA? Or both? Huh? DNA. Used for cutting DNAs. Very good. Found in source bacteria. Function protect from viral genome. So they are found in bacteria, they are produced in bacteria, and they protect bacteria from viral attack or viral genome. They can differentiate between their host DNA and foreign DNA, and thereby they cleave or digest the foreign DNA and protect them from the attack of virus. What is the way of attacking bacteria by virus? The virus which attacks bacteria are called bacteriophage or phage virus. So phage virus, what they do, they conjugate They send their DNA particle inside the bacteria and they conjugate with the genomic DNA of the bacteria or plasmid DNA of the bacteria, whatever is available, and they use the machinery of bacteria to survive, correct? To produce the viral protein. Now, these restriction endonucleases are used. Endonucleases, they are termed as endonucleases because they cut in the middle of the nucleotide. In the middle of the nucleotide, not from the either ends of the nucleotide. If they cut from the 5' end or 3' end, then we call it exonuclease. So, their endonuclease means they cut in the middle of the nucleic acid, and restriction word is designated so because their function, their digestion, their excision function is restricted to a particular site. The site which is recognized by these enzymes for the excision purpose. Excision means cutting. Excision purpose are called restriction sites. These restriction sites have got some salient features. So, for example, they are 4 to 6 or 8 base pair long, not more than that, and they are palindromic in sequence. They are palindromic in sequence. Palindromic means you read from either side, it sounds same, like madam, M-A-D-A-M. Similarly, in Bengali also there are words like Ramakanto Kamar. You read the name from either side, নামটা একই আসবে, রমাকান্ত কামার, হ্যাঁ, ম্যাডাম. So palindromic sequence, it should be palindromic, must be, correct? And these sites are, their function is restricted to site, that's why they are called restriction endonucleases. Now they can cut these sites in either of these two manner. Like this or like this. Suppose this one GATTC, this is a restriction site for EcoR1, one particular enzyme, restriction endonuclease, and this is PVU2, Roman 2, and this is cut here and this is cut after G. So they produce a sticky end and they produce a blunt end. Both the DNAs are cut at the same location. Both the DNA strands are cut at the same position. Both the DNA strands are cut in the same position. It produces blunt end, which are cut in two different positions in the same restriction site. They produce sticky end or overhang, single stranded overhang. Correct? So cutting wise, there are two types: blunt end producing restriction endonucleases and sticky end producing restriction endonucleases. Now these sites are called restriction or recognition sites. Restriction means their function will be restricted within this site, and recognition means they must recognize this site. In some enzymes, these two sites are same. In some enzymes, these two sites are different. Depending on that, okay, forget it. We'll come to this later. Depending on that, this is important, classification of restriction endonucleases. They are classified in three classes: type 1, type 2, type 3. Type 1 has got types of restriction endonucleases. Type 1, they recognize a site and they excise 100 base pair upstream to the recognition site. I mean, whatever site they recognize, the palindromic sequence wise, they cut 100 base pair upstream, 1000 base pair upstream to the restriction site. Correct? Type 2, for them, recognition site and restriction site is same, identical. So they cut within the recognition site. Type 3, they cut 24 to 26 base pair away upstream to the recognition site. Suppose they recognize this site, so they will cut here, type 1. Type 2, here itself. Type 3, little bit of upstream. Okay, so which one do you think will be more suitable for using as a tool in the molecular biology? You recognize a portion of DNA, you cut there, or recognize a portion of DNA, you cut 1000 base pair away from there. There. So on site, in situ, where it cuts. Now in some books you might find that in addition to these three types, in some books you might find in addition to these three types, there is another type new, which is artificial, not natural. Artificial type of resident nucleus is called type 2S. Type 2S. What is that? It is a modified type 2 enzyme where the cleavage site is not mentioned, is removed. They recognize the site, they bind to the DNA, but they do not cut the DNA. Why? Because the cleavage subunit has been removed. Now, all these enzymes have three types of function. One is recognition, another is correct. Now these three functions are may be located in three different subunits. These three functions may be located in three different domains. If they are separated by three separate subunits, they are called multi-subunit enzymes. Means one enzyme which has got multiple subunits. Each subunit is distinctly separate from each other. If they are somehow linked with each other, usually these recognition and modification are found together and this one is separate. If this is separate, then also it is like this. If they are linked, they are similar to what type of complex? Multi-enzyme complex. Can you give one example of multi-enzyme complex? Yes, bolo. PDH. Another one, the best, fatty acid synthase complex. Correct? Fatty acid synthase complex. Achha, PDH is not multi-enzyme complex, it is multi-subunit complex. They are not linked, they are separate. Correct? Now, follow? So, in some enzymes, these three are different. In some enzymes, they are linked, joined. In some enzymes, they are separated by this. Only two subunits. In some enzymes, the two subunits are there like this. So, this is type 1. If you remove this becomes type 2. And if you join this becomes, if you join this is type 2. And if you cut this, then it becomes type 3. But they are different. Two subunits are different, type 3. Three subunits are different, type 1. Two subunits, but they are joined, type 2, identical. Okay? No. So, what is the function of M subunit? Modification. They modify native DNA or the owner's DNA, source DNA by methylation. They have got some methylase activity. They methylase or add methyl group to DNA in particular position. For what purpose? To give it a distinct feature which is different from the viral genome. How do these enzymes recognize which DNA molecule is own DNA and which DNA molecule is foreign DNA? They recognize that by a specific methylation pattern. If DNA molecule is methylated in such and such position, then that is own DNA, my own DNA. If they are not methylated in such and such pattern, they are foreign DNA, so cleavage. Correct? Now, every organism will have some or other methylation process in the DNA. So, that is why we have a number of restriction endonuclease in a single organism. We have many restriction endonuclease in single organism. Why? Because the methylation pattern can be different from one virus to another virus. To ensure that we never miss a single foreign DNA, to recognize as foreign DNA, we have several restriction endonucleases. Followed. So the question is why one single organism has got many restriction endonucleases. Answer is this: so that they cannot, the foreign DNA cannot camouflage by polymorphism. So that the foreign DNA cannot camouflage by polymorphism. Fine? Naming of restriction endonuclease. These are the headings, these are the questions actually. These are the questions. Classify restriction endonuclease, what is restriction, define restriction endonuclease, give their types, give their naming procedure. Okay. How they are named? They are named after these headings. Named after the bacteria which produce a particular enzyme, genus, followed by species, followed by strain, and followed by order of discovery. Order of discovery. So, for example, this is an example of RE, restriction endonuclease. Echo R1, Echo R1. E stands for Escherichia coli, or Escherichia. C stands for coli. Strain is R, rough and smooth. Order of isolation or discovery 1. So, it is the first enzyme to be discovered under this series. Similarly, another example is Haemophilus influenzae, HIN D3. HIN D3. Echo R1, HIN D3. Haemophilus influenzae D3. BAM H1, Bacillus amylococcus H strain I1, BAM H1, and their recognition site GAATTC. This is the point where it cuts. Then AAGCTT, then GGATGC, like this. Their respective sites. Now, their uses in genetic engineering, they are used to cut DNA and to introduce new portion of DNA from other source. That means formation of chimeric DNA or clones. Suppose I am a dog lover, but I want all the dogs will have black color tail. So, I can order to produce dogs which have only black colored tail, and nothing is black, only the tail is black. So, if they will search for the genes which is responsible for the black tail containing dog, and then they introduce in all dogs and they produce same looking dogs. Suppose I have ordered 100 dogs. So, 100 dogs will be there with same look, same feature, right? The examples of sticky end, formation of sticky end and blunt end. Sticky end and blunt end, these are, let's go through it because this is this I will tell in another class. Another portion is there. Application of RDT, there it will come. Sometimes this is asked. Sometimes this is asked. See, how frequently would EcoR1 cut DNA? How do you do that? Maths. EcoR1 residence site is GAATTC. It has 6 nucleotide long restriction or recognition site. Each nucleotide can be of 4 types. So 4 upon 6 to the power 6. Correct? So those many possibilities are there in a DNA molecule that, but on an average, after every 4096 base pair, there is a possibility that you will get one such GAA-TTC site. Recombinant DNA technology. There are two parts. The most frequently asked question is, this is most frequently asked question. Okay. The procedure is less important, so I will discuss later. First, the application. Where do you find this application? Can you name some? Anyone from your previous knowledge? Yes, BT crops. Acha, the crops are not in medicine. Okay, I accept, but it is not in the medicine. What is related to us? Very good. Recombinant insulin. Production of recombinant insulin. Insulin are produced. Insulin has got two chains, so they are produced. So they are produced in 2 chains separately in 2 different cells. One colony of cell will produce chain A, one colony of cell will produce chain B, and after the production is done, they are joined to form the final product, that is human insulin. The use of recombinant insulin is much more beneficial and poses less side effect to the individual. Why? Unlike the previous one, which is isolated from either pig or bovine sources, they produce no allergic reaction or hypersensitivity reaction. Okay. So these are similar to human, same as human. So recombinant what else? Transgenic animals, very good. Example, knockout mice. Mice na mouse, কোনটা হবে? Transgenic animals, knockout mice. Suppose we want to do some experiment on a mice which cannot produce ferritin. So we order and they will produce such kind of mice Which will, which one is more, which one is more docile, mouse or mice? Mice is more docile, like us. Docile teachers. Huh? And mouse is imbecile, like you. Imbecile means always agitated. You hit them, they will bite you back. And mice is means you hit them, they'll go and hide in a corner. Yeah? Anyway, so they are used for experiments, animal experiments. Knockout mice is very, very useful in that. Anything else? Gene therapy. What is the disease which was cured first as gene therapy? Severe combined immunodeficiency syndrome, SCID, ADA deficiency. What else? Vaccine, very good. अच्छा हाँ लिखे दिलाओ DNA vaccine. Example? Hepatitis B, good. Then treatment of hemophilia. What is the defect in hemophilia? Factor 9 and A and B, there are two hemophilia. So they are produced artificially and they are injected in the individuals for treatment of hemophilia. What else? High proteinized milk. আচ্ছা ওটা আবার চলে যাচ্ছে অন্যদিকে আরডিটির অ্যাপ্লিকেশন মানে এগ্রিকালচারে এত আছে তাই নিয়ে এতগুলো বই লেখা আছে ঠিক আছে সুতরাং ওটা ওদিকে যাস না এটা কম সেদিকে থাক আলফা ওয়ান অ্যান্টিবিউটিন হ্যাঁ যে কোনো প্রোডাক্টই তৈরি করা যায় তবে যেগুলি মোস্ট কমন সেগুলি আমি লিখছি যেমন for neutropenia treatment. তারপরে মনে enzymes for lysis of clot. There are cases of thrombosis, blockade. So, the clot lysis, those enzymes what are used in কি বলে এটাকে during surgery মানে ওটাকে বলে ক্যাথ ল্যাব স্ট্রেপ্টোকাইনেজ আর কোন নাম আছে বল অ্যারিল হ্যাঁ থ্রম্বোপ্লাজমিনো অ্যাক্টিভেটিং এনজাইমস আর কি থ্রম্বোপ্লাজমিনো অ্যাক্টিভেটিং এনজাইমস আরেকটা নাম অ্যারিল সাইক্লেস নাকি একটা নাম বলছে ভুলে গেছি মনে পড়ছে যাও এনজাইম ফর ক্লট লাইসিস আর ইউজড আচ্ছা হ্যাঁ বলো শুনতে পাচ্ছি না লেখ না বুঝতে পারছি না বাবা এই ফ্যানের জন্য শোনা যাচ্ছে না
Cry 1AC and Cry 2AC controlled by cotton bulbous and Cry 2AB controlled by cotton borer.
কটন বোরার ওই পোকামাকড় হয়ে গেছে মানে ইয়ে তো হ্যাঁ that is another part you are talking about medicines only medicine है ना that's why I was unable to catch what you are telling huh
Nucleopolymer is controlled by bacterovirus.
any enzyme can be produced go on six almost any enzyme can be produced see when I say almost any enzyme of human body can be produced then you should have answered this what is this stands for human genome project right then making clones right these are all application for RDT plenty are there plenty any antibody any antigen can be produced any hormone growth hormone is given have DNA fingerprinting yes it is a technique of use of southern blotting growth hormone for growth correct like this plenty of there huh PCR also there PCR is another technique it's different acha fine so now we come to the second question which is less likely to be asked steps of DNA coding more or less these 6 steps are the processes by which you should go through for a cloning process. Fine. So, number 1, choosing the source of DNA. What we want to source. Suppose we want a gene which produces ampicillin. A gene which produces ampicillin. We want to clone that gene to our organism of interest. From organism A to organism B, we want to introduce. So that organism B can be self-sufficient or self-reliant regarding ampicillin production, and it will never die on application of ampicillin, because it itself can produce ampicillin, so it will not die out of ampicillin. Followed? Those organisms who can produce some antibiotics, they are by birth resistant to those antibiotics. Okay? Suppose ampicillin. So that gene, we should find out the DNA portion from the source which contain the gene for ampicillin. Then we should isolate it. Then if it is small, very little in amount, then we can multiply it. We can multiply it using PCR. If it is more, we need not. We can just isolate and perform the next step. What is the next step? Cutting of DNA. We use host and the recipient. Both the DNAs digest with same restriction endonuclease, so that they produce identical cut ends. If it is blunt end here, it will be blunt end there. If it is sticky end here, it will be sticky end there, and it will be overlapping. So they will bind. Correct? Now there will be formation of chimeric DNA. Those DNA should be mixed. Clear? Now, because it has been cut by the same restriction endonuclease, there is a big possibility that they will bind with each other with portion of that DNA, host DNA, with recipient DNA. A portion of host DNA see, this is from host, which is recipient DNA, added from the source DNA. The recipient DNA is called a chimeric DNA or a recombinant DNA, which has got its own DNA and the DNA from the other source. Right? Is it so efficient process? No, it is not so efficient. 10% of the gene DNA portion might get aligned correctly. Out of 100, only 10 molecules will be aligned the way we want it to be aligned. There is a possibility that they are aligned in opposite manner, so it will not produce the product. Correct? There is a possibility that nothing has been added. Futile. So all these possibility will reduce the efficiency of formation of chimeric DNA. Now here there are some myths like using sticky end will be better will give better efficacy, or using blunt end will give better efficacy. Blunt end. Sticky end will produce non-specific bindings because they are sticky. But blunt ends rather will produce more product. It might be reverted opposite like this, but it will produce more product, chimeric DNA, compared to sticky end. Alright. So formation of chimeric DNA is done. Next comes transformation. Transformation means how we introduce this particular chimeric DNA to the bacteria. There are several processes. The chimeric DNA goal is to introduce the chimeric DNA into the recipient organism. How? Chemical process. There are two cells. See, this is a cell. DNA of interest. I have to make it enter. How is possible? Make a door here. Correct? Make a door. Make it enter. And what is a door in cell wall? Pores. How? If we inflate a balloon, what will happen? It will swell up, so the spores will be magnified. Similarly, there are some chemicals which will introduce pores through which the DNA might enter. There is a possibility, will enter. What is such chemicals? Using calcium chloride. It will introduce pores, and through pores DNA will enter. Biological agents. Nothing is more compromising than virus as far as attacking is concerned. Virus can attack any cell. Sendai virus. Suppose there is two cells. See, the virus actually places itself between two cells and merge with their cell membrane. So both cells, now two cells become single cell. So one of this cell genetic material and this cell genetic material will be mixed. So using Sendai virus, we can do it. Last is electroporation using electric field. Sonication can also be used. High frequency sound wave. They will disperse the lipid in the cell membrane and make big pores through which DNA might enter. Okay. After all this process is done, the transformation can take place. The newly introduced DNA might form an extra genomic DNA like plasmid. It might conjugate with the host recipient DNA. Okay? In either of these methods. Whatever is the way, but transformation is done. Now the next step is, all the cells have not been introduced properly, correctly. Some of them have done it properly, some of them have failed to do so. So how do you differentiate? For that purpose, this is the most important selection of those cells which have been introduced with these genes properly. Correct? isolate them, select them and isolate them. For selection purposes, we use multiple marker systems. Multiple marker systems. Say some DNA will be given with tetracycline genes, some DNA will be given with ampicillin genes. The ampicillin genes is positive, that means they are being transferred properly. The ampicillin gene is negative, that means they are not transferred properly. Separate like that. Purify them, make single colony containing all the cells with that particular gene of target. Then culture it, produce in batch, then harvest it. Isolation, harvest it, take the product out. Now this is very simple way to explain, but each step is very difficult depending on the product you want to form. If the product is as simple as insulin, it's not so simple. Suppose you want to produce only one polypeptide, then it is simple. But if the product is made up of 2 types of polypeptide, it is difficult. Why? You not only have to produce 2 different subunits, you have to look after that they should be joined by disulfide linkages as mentioned in the structure of insulin. Then only it will function as human insulin. Then only it will function as human insulin. ADH in that sense will be much easier to synthesize, because why? It is single polypeptide chain. But insulin is much more difficult. Followed? So that is what called RDT in nutshell. Now all I will tell you, since most of the sections have been taught to you during your +2, what is important here? First is types of restriction endonuclease, their application, their naming, nomenclature. Second is application of RDT. Third is steps. Now in the steps, I did not mention one thing. Now I remember. It's called vectors. The one which carries the DNA from one cell to another. No. This transformation includes another step is choice of vectors. Vector means which carrier. What are different types of vectors in RDT? At least you remember this phage or lambda phage. Lambda phage के ये वाले. Okay. That is 5 types you can remember. Plasmid. What is plasmid? Plasmid is extra genetic DNA molecule, double-stranded, circular, found in bacteria. Do we have plasmid? We have in different form as a mitochondrial genome. Mitochondrial genome is nothing but double-stranded circular DNA. Now, cosmid is a viral genome separated by cos sites. A viral genome normally remains in association with our host cell. When threatened, when the bacteria, when they think, they sense that this bacteria where I am living is going to die, then I become hyperactive. What I will do? I will produce more and more offsprings, copies of the same genome, so that many viruses are formed when this bacteria will die. And there is a possibility that one of them will survive with my potential genetic properties. Right? So for that process, for quick packaging, the viral genome is separated by two distinct sites called COS sites. COS-SITES. COS sites. The sites between which DNA is cut and packed in a viral case. The site between which DNA is cut and packed in a viral case. Needless to say, between these two COS sites, we can remove the viral genome and replace our gene of interest. So whenever we threaten the bacteria, they will produce viral genome, virus products, but with our gene of interest. There will be no viral genome. Only our gene of interest will be there. So we will make many copies. We don't need NPCR. We can make millions of copies by COS sites. Phage-made. Lambda phage is a very big virus. Lambda phage is a very big virus. So there, that DNA, most of the DNA part is removed and replaced by our gene of interest. And then introduced into a bacteria. How? By the normal infective nature of the phage-mid. YAC, yeast artificial eukaryotic. This is eukaryotic. All these are prokaryotic. This is eukaryotic vector. Yeast artificial chromosome. BAC, bacterial artificial chromosome. Now, what is the difference between all these? The size of the DNA molecule they can transfer. The size of the DNA molecules they can just say up to 100 BP. YAC. Like this, nearby. Okay. So this can come as a short question. Vectors in recombinant DNA technology. Just name them, give some structural features, give the amount of DNA they can transfer. That's all. All you have to remember for examination is you have to write something related to the question. It is needless to say that you study one question very nicely and you score 9 out of 10 or rather 10 out of 10 and leave 2 questions. It's of no use. Rather, you attempt all the questions and get 5 marks each. It is desired. Correct? You should write something sensible, something related to the question. So at least whatever you studied in +2, try to recall, try to retain and reproduce as well in a proper manner. That is what. It's not very difficult. Only thing is, studying these subjects is actually time taking. You don't have to be very brilliant to remember all this. You have to be perseverant. You have to sit tight in one place for long hours to study this. You need not to be as brilliant as IITians. No. That much brain function is not required. Only it is required is calmness, capability to sit in one place and study. To study the same thing again and again from the same place. Read same thing, same way, again and again, repeatedly, and practice always. Do remember, more you write, more you prosper. You have to write. You read and write. You write as you read. If you think I can recall everything, I can remember, I can read and go and write, no. Exam, the mode of examination is writing. Mode of examination is writing everywhere. So practice writing, practice writing. Barbar bolchi. It is a writing which lacks. Write, write, write. That's all. লিখে প্র্যাকটিস করবে, যা আনসার হবে লিখে প্র্যাকটিস করবে। অ্যানাটমি অ্যাজ এ এক্সাম ইজ অ্যাপ্রোচিং, অ্যানাটমি ছবি আঁকবে লিখবে, ছবি আঁকবে লিখবে, পয়েন্টস লিখবে, বায়োকেমিস্ট্রি পয়েন্টস লিখবে, পাথওয়ে লিখবে,